Welcome. My name is Nathan Babcock, and I'm happy to present to you today our findings regarding prospects for magnetic sensing in cryptochrome based on the radical pair model. This research was carried out in the Catning Lab at the Living Systems Institute with the Department of Physics and Astronomy at the University of Exeter. In this work, we find that electron-electron dipolar interactions pose a challenge to established models of the radical pair mechanism of magnetoreception in the protein cryptochrome. The radical pair mechanism for magnetic sensing was developed after it was shown that birds, such as robins, like the cute little one shown here, have a visual sense of the inclination of the Earth's magnetic field axis, but not its polarity. This means that birds can effectively see the angle that the Earth's magnetic field makes with the ground, but they cannot distinguish between its north and south poles. Naturally, this sense is believed to assist migratory birds in their seasonal journeys towards the equator in the fall and back poleward in the spring. In the 1970s, Schulten and co-workers noticed that this peculiar avian faculty might be rationalized in terms of a radical pair reaction mechanism in which the formation of the reaction product is sensitive to the orientation but not the polarity of an applied magnetic field. Without a known receptor to mediate the avian magnetic compass sense, this theory waited in the wings, so to speak, until the discovery of cryptochrome by Ahmad and Kashmir in the 1990s. In 2000, Ritz and colleagues formulated a hypothesis that a radical pair reaction may mediate a magnetically sensitive effect in the photoreceptor protein cryptochrome, which has been the leading hypothesis until now. The identities of the radicals in question remain uncertain and are fiercely debated, but two basic proposals have gained prominence. One proposed model relies on a flavin tryptophan radical pair generated in a photoreduction reaction, as seen here. The other model assumes that magnetosensitivity results from the dark state reoxidation of the fully reduced flavin cofactor by a free radical such as superoxide. In any case, these models of magnetoreception in cryptochrome have garnered substantial attention over the last 2 decades as scientists have demonstrated magnetic field effects in cryptochrome molecules in vitro. However, the majority of the relevant theoretical work so far has focused on phenomenological toy models with limited practical applicability. These models have often neglected to include all relevant physical effects, such as electron-electron dipolar interactions and electronic spin exchange. The substantial influence of these effects tend to break the fragile energy degeneracies on which the radical magnetosensitivities rely. The conventional radical pair mechanism has a strong focus on the radical's interactions with the local magnetic nuclei and the applied field. However, the mechanism based on electron nuclear couplings can be hampered by other electronic interactions. It was hypothesized that the mutual cancellation of electronic dipolar energy, D, and exchange energy, represented with a J, could produce circumstances in which the two forces partially eliminate each other. Such a JD cancellation effect would justify the assumption that the two effects partially cancel and can be treated as negligible, which has often been taken for granted. These figures show the angular dependence of the magnetic field effect for the conventional radical pair mechanism alone, in red, or in the presence of electron-electron dipolar coupling, in blue, and finally with or without electron-electron dipolar interactions and exchange to match the supposed cancellation conditions, with the positive matching condition shown in yellow and the negative one shown in purple. As you can see, our model predicts that JD cancellation fails to restore the directional sensitivity in cryptochrome. After the conditions which were anticipated to restore magnetosensitivity to these systems failed, we felt motivated to carry out an exhaustive test of all possible exchange coefficients for the two hypothesized radical pair systems, as shown here. Although this exhaustive search revealed the richly complex dependency of the field effect on the exchange coupling, it did not support a tractable model of JD cancellation in molecules like cryptochrome. This prompted us to consider whether the magnetic field effect of the radical pair model might need to be revised in an alternative approach. The theoretical physical basis for the radical pair model lies in the hyperfine coupling between nuclear spins and the pair of initially entangled radical electrons. Previous work by the Catenick group showed that a third radical could supplant the role of the nuclear spin in the usual radical pair picture. Thus, radical pair model systems may be conveniently generalized by including a third free radical in the picture. This alternative three radical formulation depends on the simultaneous interaction of a trio of radicals and thus their relative geometries, interaction strengths, and symmetries. Magnetic effects are predicted even for rather remote locations of the unreactive third radical, which suggests the name bystander radical, and which I'll use henceforth. We hypothesized that the additional flexibility originating from the variable location of the bystander may give Mother Nature an opportunity to tune interaction strengths to meet the specific demands of the magnetoreception problem. Our simulations of an extensive range of bystander positions indicate that substantial magnetic field effects can emerge from a system represented by just 3 electrons, including the primary radical pair and a bystander radical. Without even considering nuclear hyperfine interactions, we found that sizable magnetic field effects result even for radicals placed far apart and could be enhanced by the appropriate tuning of an exchange coefficient, as shown on the left. By including nuclear hyperfine interactions in the system, as we see on the right, we found that the possibly intrusive magnetic field effects predicted for most bystander locations without even having nuclear coupling can be largely suppressed. But significant magnetic field effects can still be obtained for bystanders when they're placed in specific locations around the protein. This came as a surprise. For one flavin tryptophan model scenario, which we see on the right, simulations indicated that we might realize an enhancement of a factor of 20 compared to the directional anisotropy of the radical pair model when this model includes the unavoidable electron-electron dipolar interactions. This prediction prompted our idea that the cryptochrome itself might have dedicated bystander docking sites to enable or disable effects on demand. We found very few regions where the placement of a bystander radical produced sharply peaked magnetic field effects, and these typically were close to one member of the primary radical pair or the other. The positions of these optimal bystander locations are shown here, nestled into the protein tangle, with insets revealing close-ups of the bystander sites near the semi-transparent protein surface. The close proximity of the optimal positions to one of the primary radicals prompted questions about the role of radical bystander spin exchange, which can become substantial for radicals in close proximity. The figures here show the scale of the magnetic field effect that is predicted when the third radical is optimally located with respect to the flavin tryptophan radical pair, as we tested an exponentially large range of radical bystander exchange coefficients. As you can see, our results are robust against even very large exchange effects between a radical and a bystander, and produced an enhancement even around one particular value. In conclusion, our findings indicate that electron-electron dipolar interactions in radical trio model systems can produce large enhancements over bare radical pair-mediated magnetic field effects. Yet, these interferences between electron dipolar and hyperfine interactions present some insurmountable problems for a magnetic sense using a lone radical pair. However, in the three radical system, they may instead carve out narrowly defined, magnetically optimized locations which could designate docking sites for radicals on the protein surface. This work was supported by the EPSRC, the University of Exeter's High Performance Computing Facility, and several of our insightful colleagues. Thanks very much for tuning in, and have a great day.